Hi, welcome to Detours, Understanding Acquired Brain Injury. And this is our second of the first chapter, our second unit for neurogenetics, things that can go wrong, specifically with chromosomes. We already have talked about aneuploidy, where both in autosomes and sex chromosomes, there are either too many or too few chromosomes, which lead to obvious problems due to the sheer number of genes that are either doubled or tripled or completely deleted. Only one deletion is survivable. We mentioned that about Turner syndrome. But what other kind of anomalies exist besides aneuploidy? There are two other common chromosomal disorders which are related to a problem with the chromosome itself, and that is a ring chromosome and a translocation. Okay, and a ring chromosome is exactly what it sounds like. Sometimes, especially with smaller chromosomes, the two ends get stuck together. And a chromosome forms into a physical ring. Looks like this. A ring chromosome may also form when a chromosome breaks into two parts, and the broken pieces then re-stick together to form a ring. Remember, there's an electrical charge on the chromosome, so it's like chemically they become bonded. Obviously, it's difficult to decode a ring for the RNA to read and decode it into proteins. Inevitably, genetic material will be lost. What's most likely to be unreadable is what's the material that's at the ends that stick together to form the ring. There are specific chromosomes, because it's the short ones, that are most likely to form ring chromosomes. So a ring 14 chromosome will give you intellectual disability, seizures, developmental disorders, and delayed growth. I think you may see a pattern here. Ring 20, intellectual disability, seizures, behavioral disorders, delayed growth. Ring 22, intellectual disability, poor muscle tone, delayed growth. Ring X, sometimes when this happens, you get Turner syndrome because it's the same as a deletion. There are circumstances, and this can come from either side, because usually this will happen after fertilization and things like that. But we will discuss the difference between the outright deletion and the ring chromosome, because it can be partially read, and therefore it's not a complete kind of Turner syndrome that a deletion represents. Okay? The other thing to keep in mind is a ring chromosome is very unstable. They tend to break easily during cell division, and this can lead to the loss of even more information. So it is an important thing to keep in mind. The other common problem is what's called a translocation. The exchange of genetic material from one chromosome to another. Translocation can lead to non-survivable conditions, permanent disabilities, serious disease, or no effects at all. Obviously, as you might imagine, this comes from where this happens. Where you might say, well, no, nothing at all. If the gene is read from the other chromosome, then you might be protected against this. For example, in a female, where her other X is the one that is being read, if there's a break in the non-reading X chromosome. Important thing to keep in mind about that is it's still possible that broken X may be the one she passes along, or damaged X. We will talk about fragile X later, but there are others as well, because not every gene is turned on. Okay, so two types of translocations. A Robertson's translocation. Different chromosomes fuse due to the loss of their centromere, which binds them up. Remember how in the previous set where we talked about genetics in general, genes and chromosomes, that they're bound together. and they fuse their long arms, the Q arm, which is the longer end, and they fuse, break off, and just decide to go for a trip, I guess, inside the cell. Very serious, that amount of loss, because usually the longer leg, if the Q arms fuse, or if the P arms, the shorter arms, break or wander away, and then you end up with some strange joined, non-functional or partially functional. So, Robertson's translocation. Certain chromosomes are likely to do this. And they're related to what we had spoken about before when it came to aneuploidy. Chromosome 13, 14, 15, 21, and 22. So you can have conditions that look similar, either by a duplication or by a loss. Yes, we will discuss. Due to the chromosomes involved, there's such a thing as translocation Pitot syndrome and translocation Down syndrome. Essentially, even though there may only be 2 chromosomes of the 13th or the 21st chromosome, there may actually be 2 fused long arms which act like an extra chromosome. They double up the equivalent proteins and end up being decoded as though there were two to be read instead of just one. Remember from our cells and proteins video how we said that the body will just read one copy. But if there are 2 floating around, one misfused and the other that is the copy that's being read, you'll get a double up. And a lot of the proteins that will cause symptoms of Down syndrome or Pitot are on that long arm, which is misfused. All right. Another type of translocation is called reciprocal translocation. And this plays a role in cancer, and something you may have heard or not. This type of translocation involves an exchange of segments between 2 different chromosomes. They may be balanced or unbalanced segments, so short and long arm or long and long arm. Leukemia can be caused by a translocation. The Philadelphia chromosome, in some balanced cases, translocation. So you're talking about it breaks from one segment, fuses to another. This is the Philadelphia chromosome, which is associated with the development of cancers, leukemia specifically. But that's what the Philadelphia chromosome did. It is a reciprocal translation. And because of misreading the information, it leads to some of those errors that are necessary for cells to become cancerous and to reproduce. They need to have certain characteristics. We will discuss that when we do get more into detail on cancer biology. But those errors are due to the reciprocal translocation and fusing of the genetic information. And obviously, if you need to read the entire segment and it is fused, some of it will inevitably be misread, and it will lead to those kind of errors in the production of, for example, white blood cells that are different from the wild type, the standard type that develop into cancerous cells. All right, so something else to keep in mind is an isochromosome. This happens when a chromosome divides horizontally instead of vertically. This leads to if it's right through the middle, remember where we mentioned the centromere, which bonds in the middle? So if it breaks horizontally, this may lead instead to a duplication of the short or the long arm, and you lose that information that is necessary from the part of the arm that did not duplicate. It can behave similarly to a translocation, but the problem with the translocation is the break may be anywhere along that entire P or Q arm. But an isochromosome is in the middle where centromere joins the two. So you get an entire P or Q segment. Again, go back to our genes and chromosomes if you have questions. Isochromosomes are seen in Turner syndrome and in many cancers. Okay, so an inversion. An inversion is when a chromosome breaks at a segment or part of an arm, and then, as you might imagine, gets flipped and joins, either back to the original chromosome or with another one, especially if there are multiple breaks. But typically, it's itself. So, you've got your chromosome here, and the long or short arm breaks and reattaches, and this is called an inversion. And obviously it would lead to those genes being misread because they're upside down. There are several different conditions associated with that, but we will be getting into more detail, but several of these are inversion-related neurogenetic disorders. Sotos syndrome, we'll talk about that one. Williams-Buren syndrome, Wolff-Hirschhorn. Those are all due to inversions, where the gene, where part of the chromosome got flipped and read upside down. The other thing to talk about, finally, for chromosomal type disorders is fragility. Chromosomes may have sites that are what is described as fragile. And you're familiar with the most famous one, Fragile X, but there are other sites of fragility. And I do want to talk about that shortly, but these sites are weak, are sites where the bonding is not as effective, and you could see it under an electron microscope or when examining it, and they're more likely to break off. They're weak. They're a lot of repeats and sites with several errors on the chromosome. And so, yeah, these are things to also pay attention to, because they can, but not always, lead to genetic errors. We're discovering that it was more than the Fragile X. We're discovering now these other sites. Okay. Obviously, we'll be talking about these disorders too.